There are different approaches. One is based on biology, which means if a patient has an IDH mutation, it seems to be reasonable, obviously, to use IDH inhibitors in this patient. Same is when we have patients that have overexpression of tyrosine kinases or mutations of FLT3 or KIT, for instance. Those patients might benefit, as we've seen from the midostaurin trial, the RATIFY trial, that targeting mutations by tyrosine kinase inhibitors such as midostaurin is beneficial for those patients. And apart from those biological rationales, there is still a lot of trial and error as well going on, meaning using drugs in certain patient subtypes we think who might benefit, and then looking at early efficacy readouts in the clinical trials to see whether actually all those patients benefit or whether it's only a subgroup. And very important when it comes to early readouts of clinical trials is progress in the detection of measurable residual disease, MRD. And there are two approaches, like molecular biology-based approaches, such as PCR or next generation sequencing, but also flow cytometry based MRD assessment.